so early. Presentation on? Do I need a microphone? What? All right. Does it work? Does it work? Test, test. Does it work? Does it? No. Oh, okay. I'll just. Okay, so I'm just going to try to talk a little bit louder so that you can all hear me in the back. So originally I was going to talk about stress in dandelions and how it affects patterns of DNA methylation, but I only have some very preliminary data that I will show you in the end. So instead I just like to talk about measuring epigenetic variation in non-model species using a technique that we've developed in our lab. It's called epiGBS, so I'll take you through that. So, as you all might know, DNA methylation, or epigenetic mechanisms in general, are very interesting. In plants, they've been known to cause some different phenotypes. Here we see Linearia vulgaris. Actually, Linnaeus himself described this caloric plant as a monstrous mutant. And little did he know that it was actually an epigenetic variation rather than a genetic variation. So, you can imagine that if a flower type really differs by this much, this can have an effect on speciation or on the way that plants interact with their pollinators. So this is one of the few examples which is known. So the way we can think about epigenetic variation really is in the interplay between genetic variation and selection and adaptation. Selection, of course, occurs on the phenotypic variation of a plant, and components of heritable phenotypic variation are determined by genetic variation and potentially also by epigenetic variation. So that's why I think it's really interesting for ecologists and evolutionary biologists to sort of think about epigenetic variation. So a very simplistic model of DNA methylation could be genes that are switched on and switched off. If a promoter is methylated, then typically a gene is switched off. If a promoter is not methylated, the gene is switched on. This is of course a very simplistic view, but it gives you an idea of what can happen with DNA methylation. So another conceptual point about, sorry, the arrows just moved, but about the difference between genetic and epigenetic changes is that a genetic change is generally a one-way street. So you go from an original gene, you get a mutation, and if the gene is broken, it's very unlikely that that mutation will happen back. Whereas with an epigenetic change, you have the potential of reversal. So you can have a methyl group that can have a change and that can revert back to the original state. Microphone? Do we need it? No? I'll just. I don't need it. Okay, I'll just continue without the mic. So you can think about heritable epigenetic variation in two ways. Basically, we can think about it as a response to environment, like phenotypic plasticity or maternal transgenerational effect, but also in the sense of variation that's being generated. So heritable epigenetic variation can also have a random component, and then it can be acted upon by selection. So one of the very cool experiments that was done in the lab of Frank Johannes and Colo was the epirails. So epirails are synthetic lines of Arabidopsis in which there is a line which is deficient for a methyltransferase and a wild-type plant. And when you cross them, what you get is essentially a pattern of normally methylated and unmethylated DNA. And this actually has a very drastic effect on the phenotype of the plants. So that shows that epigenetic variation can, in principle, also have an effect on the phenotype of plants. But this is, of course, in a lab in a highly artificial situation. Our question is, does it also play a role in nature? Some more work that has recently been done on this is work on epigenetic mutation accumulation lines, also in Arabidopsis, in which they followed what happens to the methylome of Arabidopsis through time. And then what they found is that, yes, phenotype also start to differ. There's like divergence. And the spontaneous epimutation rate of cytosines to methylated cytosines is 100,000 times higher than that of SNPs. So there is a potential there for very rapid change, and this is, I think, for ecologists, a very interesting notion to take care of. So here you see, like, after 20 or 40 or 60 generation, here there's the CG methylation divergence. That's between, you have a mother plant and then you follow it through some generation, and then you see that basically CG denucleotide methylation patterns start to diverge. However, that pattern is most drastic in genes, whereas in transposable elements it's mostly conserved, consistent with the hypothesis that transposable element must be controlled by DNA methylation. So it shows that the dynamics of DNA methylation polymorphism and the changes therein depend on the context of the DNA. So I'm working in dandelions together with Koen Verhoeven in his lab, and we're using common dandelions as a model for epigenetic studies. Why? Well, dandelions are ubiquitous. They grow pretty much everywhere in the world. And there's the sexual dandelions that live in France. And when you cross a diploid sexual mother with a triploid father, you get a triploid apomate. And that apomate can then give rise to new offspring that's essentially a clone. So you have mother, daughter, mother, daughter, and genetically it doesn't change very much, yet these plants are able to colonize a wide variety of habitats, which is why we're thinking like maybe epigenetic variation could play a role in the adaptation of this dandelion to new habitats. So this is just a picture I pulled from the internet that shows some observations of dandelions all over the world. I'm pretty sure it's not complete, but it gives you an idea that it has a pretty cosmopolitan distribution, where it's originally from Europe, especially North America. It's been pretty successful in colonizing mainly disturbed habitats. So this is just like a cartoon-like representation about how we measure epigenetic variation, in which I draw the ecologist and the molecular biologist. And the current state-of-the-art for ecologists, unfortunately, is a technique that I think was developed about 20 years ago. It's methylation-sensitive AFOP. What it does, basically, you have your genomic DNA, and you have some restriction site. You digest your genomic DNA in this restriction site, you end up with fragments. You put these fragments on the gel, and because you have a methylation sensitive, sorry, this one is sensitive, this one is unsensitive. When you have methylation, this one doesn't cut, yet this one does, and then you get this 1-0 binary pattern, which basically gives you a qualitative measure of a quantitative state and tells you something about CpG methylation at a number of anonymous loci throughout the genome. So it does help you to get an idea of is there epigenetic variation in any given population, but it doesn't allow you to appreciate what it actually means, which is why we thought we should develop something else. Now, Bisulfite sequencing, which is commonly employed in model plants, involves conversion of DNA, in which unmethylated cytosine, here depicted in blue, get converted to uracil after bisulfite treatment, and then if you PCR amplify, that uracil gets translated into a T. So essentially you get CT polymorphisms that are not nucleotide polymorphisms, but that depict DNA methylation variation. So this is what it looks like if you have a genome and if you do whole genome bisulfite sequencing. Basically for every single cytosine you can say, well, this is the quantitative DNA methylation state for that individual nucleotide, and this is something we'd like to get at also with non-model organisms. So what you can do with this, what the power that this gives you is if you can look at individual dinucleotides, obviously, let's say you have two groups and you see here that this group is methylated and these groups are not very methylated, then you can say, well, is this significant or not? And often when you look at a single nucleotide, it won't be, but if you look at an entire region, it might be. And more importantly, if this happens in promoters, or if this happens in a gene that you know has a certain function, then you can say, well, we're seeing DNA methylation differences, and they occur in this and that gene, and that can actually lead to more informed hypothesis and answers to questions that you might have. So we decided to go with genotyping by sequencing and sort of make the transition from genotyping by sequencing to epigenotyping by sequencing by incorporating bisulfite treatment. So the way genotyping by sequencing works is the same with methylation-sensitive AFOP. You have your DNA, you digest it, you have your fragments, you ligate barcoded adapters, and you PCR amplify, and then for different samples you get different fragments, and then you can observe SNPs in them. So GBS is very successful. It's applied in many normal species. It's cited very well, and everyone seems to love the method. I think it's mainly because it's very flexible, it's easy to use, and it doesn't require a reference genome. So epiGBS is very similar in concept. So if you have a piece of DNA here, and you have methylated cytosines here in orange, and you have blue cytosines here in blue, the unmethylated cytosines in blue, when you digest, obviously, you get a fragment, and then when you do bisulfite sequencing, these Cs get converted to uracil after PCR amplification to become a T, and then you can see, if you would combine bisulfite sequencing with normal sequencing, you can see, okay, so here we have a CT polymorphism, so that must mean that there is a methylation polymorphism, yet here the C remains intact, so that means it must have been methylated. So that's the way it works in a nutshell. It's a bit more complicated. I'm not going into all the technical details, but basically we're able to do this highly multiplex DNA methylation genotyping by sequencing for up to 10,000 loci, and the output of our pipeline generates this sort of heat map in which you see methylated nucleotides, you see CG dinucleotides, and you get like the sequence that it was derived from. So it allows you to get a nucleotide level representation of DNA methylation in up to 96 samples in one Illumina lane of many different species. And you use the barcode to basically say, well, okay, this individual fragment belongs to that sample. It's very straightforward. So the idea is that it should be somewhere in between methylation-sensitive AFOP, whole genome bisulfite sequencing. I won't go through all the specifics, but basically what we say, well, we want something better than MSAFOP, but we cannot afford whole genome bisulfite sequencing, and it's also not very practical. If you don't have a reference genome, you can forget about whole genome bisulfite sequencing. So we validated the approach. Well, it was important to see, okay, how well does this actually work? So it's similar to existing reduced representation bisulfite sequencing techniques, but it's a little bit more advanced, and it has slightly more flexibility. So what we've done to validate it is basically to look at the Arabidopsis line of Becker et al. And so we've obtained some seed materials, and then we've compared CG methylation in line 30 with line 31. So basically mother-daughter offspring pair, and then we looked at a subset of loci, so it's about 3,000 fragments, about, 12,000 CG positions, for which you can look at, okay, so mother-daughter. And basically, the inheritance is pretty strong. It's about a 95% correlation that you get when you plot them. And then we've done the same for the material that we've derived from whole genome bisulfite sequencing with the material that we've obtained from this epiGBS, and you see the exact same pattern, meaning that the accuracy of the way we measure it is as accurate as whole genome bisulfite sequencing. Now, we successfully performed it on many species, mainly plant species, just as a proof of concept to see, okay, can we actually do this in a non-model or a model species, and can we say something about DNA methylation patterns? So one of the first results that we got from this is this picture that shows CG, the fraction of methylated cytosines in CG context. So methylation here we define as being methylated more than 5%, and we have some coverage threshold. It needs to be more than 15 times, I think it was. But what you see here is that there's quite some dramatic variation in CG methylation with human DNA. CG nucleotides tend to be methylated most of the time, whereas in Daphnia, there's almost no CG methylation, although it is there. And you also see that Arabidopsis and Terexica have very low levels of CG methylation compared to some other plant species, with Allium corum being also very low. And if you look at non-CG methylation, you also see that there's quite a lot of variation, although typically, as the threshold we've used here is 5%, there's 2% or less methylated nucleotides in this context. So, onto the system that I wanted to test this on. So we're working in dandelions, and we're working in a system in which we've done an experiment of stress, and then to see, okay, if you apply stress to the lineage, does this have an effect on the epigenetic variation that we observe, and is this transferred to the next generation, yes or no? Right now we're only focusing on the stress effect. So jasmonic acid is a plant hormone that's typically released after the plant is being subjected to an attack by herbivores. So there's a pathway, and then if jasmonic acid is involved, and then this jasmonic acid triggers all sort of defense responses. Okay, I'm done. Oh, okay, I should be done. All right, so really there's not too many results to show here other than at this point we didn't really find any significant differences in DNA methylation levels between the jasmonic acid and the control plants. With that, I think I should end maybe answer one question and then we should move on. Yes.
Any way to look at like single-stranded methylation versus double-stranded?
So yes, we are able to separate the Watson from the Crick strand. So I don't know if that answers your question. So methylation, it has an effect on the cytosine, but the guanine on the bottom doesn't get affected. So when you have the Watson and the Crick strand, you have information on both, then you can distinguish between SNPs and DNA methylation variation. Does that answer your question? Yeah. Yes. Okay. All right. So we should continue.